so we continue with with Volker from Bern about talking about research tools.
So thanks a lot, Sultan, and it's nice to see you again. I will also take you today back in time. And many thanks to the organizers. I also have to say many thanks. A year ago, we organized the nidovirus meeting, and the UK ICN was the first to offer sponsorship and support, which helped us a lot. So thanks for that. And of course, this place here is just super nice. So I want to take you a little bit back in time, as I already mentioned, into tools we have already developed during the years for feline coronaviruses, but also give you a little bit of outlook what could be done and what systems could be available to achieve which goals or aims. So that a little bit of discussion maybe in the end what might be reasonable. If we think of a transmissible FIP virus, it might be reasonable to get back to a vaccine maybe. And treatment is obviously very, very successful, but expensive. So might not be solution for every case. So if I go back in time, I also have to mention briefly how we were working at the time. So this is the technology we had more than 20 years ago. Sultan, you may know these kind of slides. Julian, you also know them very well. So this was the time when we were discovering ways and elaborating on, systems to clone coronaviruses. So essentially this part here in 2000 when essentially 3 systems for cloning coronaviruses were successful. And we all know this was a lot of suffering for many years to really find a way to bring together these 30 kilobases in one piece of DNA. And not surprisingly, this was done with very unconventional systems. So we all were driven a little bit by early reverse genetic systems that had a simple plasmid, and you just cloned it and then you recovered the virus. For coronaviruses, this was much more complex. And there were essentially, I remember the nidovirus meeting in 2000 when 3 systems suddenly were presented that were developed or were successfully established in almost at the same time. So one is driven, what you see here on the left, mainly by Sultan in Luis and Juanes' lab, based on a bug system for TGV. Another one was made by Ralph Barrick's lab with a set of overlapping DNA fragments that were then ligated. And we went to use vaccinia virus as a non-prokaryotic cloning system to incorporate the cDNA of a coronavirus. All 3 systems were successful, and they served us for many years to study basic questions on the biology of coronaviruses, gene function. So you could now, for the first time, knock out certain functions and see what the phenotypes are. And nevertheless, we were always a little bit not unhappy, but it was just not fast enough. So making a mutant took several weeks, and essentially you could do much more if this process could be shortened. And we started a couple of years ago when Jörg Jores, so he's a bacteriologist in our faculty, when he joined our faculty, and he brought a system with him that is based on cloning of DNA in yeasts. And we thought, why not giving it a chance to see whether this can be beneficial for us. And we were at the time in a European consortium, in a European project called HONORS. So that was a pandemic preparedness consortium And our task was to make a reverse genetic platform that is really fast and applicable to many viruses. So this was done essentially by Tao and Nadine at the time in our lab. And the system is very simple in a way that you just need to have DNA fragments. You see here our first try with mouse hepatitis virus that we already had cloned and inserted a GFP to easily monitor infection. And we just used this as a template and made simply PCRs. Each PCR was about 3 kb long, and we had overlaps between fragments and overlaps to a plasmid backbone. And then you don't need to do anything else but just put these fragments into yeast. So you transform yeast, and then yeast will do you the favor to assemble this nice plasmid in the right order. You get your cDNA stick together, and from there on you can then use this DNA as we did before to make a copy, an RNA copy, which is essentially then the viral genome, put it into cell and harvest the supernatant. And this was extremely successful. So we randomly just picked 2 clones of this first approach, and they were both working. So this was the day when we discarded our loved and hated vaccinia virus system and switched completely to this one. In 2019, so we further explored until then, is it applicable to other coronaviruses? You see here on the list, MERS coronavirus, also with a GFP, 229E, HKU1, so other coronaviruses, a MERS isolate from camels, and even beyond Zika virus, beyond coronaviruses, the Zika virus could be easily cloned, and also hepatitis or RSV, which was quite interesting because we just used conserved regions for our consensus primers and amplified 4 fragments. And in the beginning, we just knew from the diagnostics that this is RSV. We had no idea what type of RSV, so it turned out to be RSVB. And this showed that we can also use this system going from clinical samples directly to clone the virus. And everybody of you know what happened then in 2019. We had this outbreak of SARS-CoV-2 in China. The first cases were reported in early December. And this, of course, was for us the starting point to say, okay, now we have developed this system. We want to apply it and clone the SARS coronavirus as fast as possible. So we received information about the genome sequence. They were released in January 10th and 11th. And then over the weekend, we designed the fragments and ordered essentially the fragments to be synthesized. It took 2 to 3 weeks until we received the fragments, and then the assembly of the clone and the rescue of the virus, that only took 1 week. So in February 12th, we obtained the first recombinant virus in our lab. And just to remind you, at that time, SARS-CoV-2 was still only isolated in China, so it was not possible for anybody to get access to the virus. And the first outside China isolation was on January 28th in Australia. But from there on, you have to grow up the virus, and then you have to do all the shipment paperwork. So it takes quite some time to really receive something. So that was for us also nice to see that we can really use this system in a pandemic situation to very, very quickly get the virus into the lab and try and start working with it. So going back into time again, so this, when we look at feline coronaviruses, also what we can do with those clones. This is a paper we had in 2008. So together with Gary Tekes, we decided at some point, okay, this feline coronavirus is so interesting, the disease is so difficult to tackle. It would be really good to have an infectious clone. We knew, of course, that getting good isolates for propagation in cell culture is a real challenge. And what you get is always not really phenotypically reflecting how the virus may have looked in the cat. So, but nevertheless, at this point we started with a type 1 feline coronavirus clone based on the strain black. And you see here still the laborious way of assembling the genome into one long cDNA. But with this, we could also then introduce a reporter gene like GFP, and we could also look for things like what you see in the images, monocytic cells or dendritic cells, whether they can be infected. So that was really nice to see that this system worked. Gary, unfortunately, is not longer in academia, so but he nevertheless, before he left, so he's now at Elanco, he further developed the system, and he was interested in getting a clone from a real field isolate, but from an enteric virus, so not a FIP virus, and he was very successful in that too. The only bottleneck here is, of course, that once you have assembled the clone, you cannot just put the virus that may be released from the transfected cells onto a cell line, because there is no cell line to really propagate those field strains. But he circumvented that by going with the supernatant directly into cats, and so that the virus is infectious and is shedded over several weeks in feces. So by this he established sort of a model for the enteric infection. And he also then went on to, because there is no good cell culture system, to try if organoids will work. Here's one example from his recent work where he collaborated with Megan Stanifer and Steve Boulon. They are now also in the US. They have been in Heidelberg before, and they checked if feline gut organoids might be susceptible to infection with this field strain. And for this, they also introduced a GFP so that they can see the infected cell, and they could show that this is indeed possible. So that's also maybe a way to look what are the target cells in something like an organoid that may contain the real cell types that are that we find in the gut of cats. What we did, and this is a paper we just published now, but the work is also going back to a collaboration with Gary in Gießen. Beside the infectious clone for this field strain, he also generated a replicon system based on the same sequence. So what he did here is to essentially delete all the structural genes and accessory genes except for the nucleocapsid, and the 7B is still in there, and add a GFP. And what we also wanted to have is a selectable marker. This is shown in the sequence here. So we have a neomycin resistance genes introduced in a way that by translating the open reading frame 1A at the beginning, at the NSP1, NSP2 cleavage, we get expression of this neomycin resistance gene, and thereby can select for replicating virus, or in this case, replicating replicon. So what you get then when you transfect this RNA into cells, you can select for the replicon RNA, and the surviving cells, they should contain the replicon. And you can see that then, and they should also express the eGFP as a subgenomic mRNA. That's why we see them as green cells, and the nucleocapsid protein. And this is what you see here. So not all cells are green, but a substantial fraction of them. And you can then easily measure the green fluorescence, either intensity or number of green fluorescent cells. You see here also on the right side the treatment. This was done with GC376. And you see with the different dosages that the GFP cells or the GFP positive cells, they slowly disappear. So that was then a reasonable thing or system to look for other inhibitors, and we will also employ this system later on, as I will show in a few minutes, in combination with clinical trials as sort of a way to connect the clinical studies with lab work to monitor what's going on. And here you see a couple of examples with a GC, the essentially Paxlovid or remdesivir treatment. All are very effective, and our system reacts in a way that we were hoping for, that the GFP cells, GFP positive cells, they disappear. So that's a very easy way now to assess those antivirals. You can also, sorry what I forgot, you can also ask, which is on the bottom right side, is there any benefit in combining antivirals? So here, for instance, remdesivir and Pro inhibitors, whether there's a synergistic effect or not. So where do we go from there? With a replicon-based setting, we can then now with feline coronavirus 23, we could think about making virus-like particles. So we could take those replicon-containing cells and transfect, in addition, two plasmids encoding for the missing structural proteins like spike or ME, and then obtain particles in the supernatant. And if those particles will infect essentially target cells, we would see them green fluorescent as green fluorescent cells, and that's maybe a good way to find the target cells of this new virus in a setting that is very flexible, because you can imagine you can easily modify the spike or exchange the spike. That's one thing. I also want to show what we started recently, also in Bern, a clinical study, treatment study, where we essentially used the same compounds as everybody else. But for us, it will be good to gain experience, and we would also like to connect that then, as I mentioned, to the lab work, because what we have not seen so far, or also in the literature, there's little evidence of resistance. So is the virus acquiring resistance to drugs? And that would be nice to connect that to the lab work and introduce the mutations we see into our clones and see whether they may become a little bit resistant. So what we did so far is in our small animal clinic in Bern and several private clinics that we recruited cats. We went through the procedure that we have seen in the other talks also, that we are sure or almost sure that they have FIP, and then we treatment them for at this time with remdesivir and GS for 12 weeks, and we connect that to studies on, of course, the detection and monitoring of the viral loads. We also do sequencing, and we extended that now to sequence also non-FIP cases. So several breeders are happy if we sequence what their cats are shedding in the feces. And we already have one case that turned then into a FIP case. So that would be a very nice way to use also the sequencing technology we have now developed to maybe get to a couple of enteric and FIP virus cases or sequences. And of course, we would like to see if mutations occur during treatment or when they relapse, how the virus is looking like. So just a brief outline. I mean, this is very basic. So as I said, we want to get first some experience, but we have already started with that at the beginning of this year, as I said, mainly with remdesivir and GS. So up to now, 26 cats were recruited and 24 cats received the treatment, and 19 are still under treatment, and 4, as we have seen in the previous talks, died also very early on. So these were really terminal cases almost, or in one case it was actually not FIP in the end. So these are the first steps we are going. Maybe a few words also on the sequencing. So we started off because with the idea to really get an overview what is around in Switzerland. So because we have no idea about diversity, what strains are circulating. So you see here in green, these are representative viruses from GenBank to illustrate, let's say, the diversity we have overall. And in red, you see the Swiss sequences with the FIP cases. These are the squares, yeah. And in circles, you see sequences from a breeder. These are non-FIP cats. But as you see, there's already one square. So this is the case that turned out to develop FIP later. And you see that the sequence space we have in Switzerland is pretty wide. We also have type 2 and type 1. So we will continue on that to get an overview what we have here. All right, with this I'm essentially at the end. So this is one of the more latest group photos. We have Kim here in the middle, who is doing the replicon work and the molecular virology work. Alejandra in the front is doing the clinical study, so she is very much engaged also in the sequencing of the field viruses. And in the back you see Hans-Peter. He's essentially a lab technician, but he is a full-grown and experienced scientist, so he's doing all the sequencing work and phylogenetic analysis. So what can we do with this? I think the molecular clone, one thing, as I said in the beginning, maybe a vaccine might be of interest if there is an FIP virus that is transmitting well. Then, of course, for basic gene functions, we still have not a good idea what particularly the accessory genes are doing. The replicon cell line, we think, is quite good for assessing inhibitors, but also later on to look at resistance if it may develop. And the virus-like particles are probably very helpful also to find a cell culture system that is good, but also in the context of the organoid system. And collectively, these three parts may give us some idea of the target cell tropism. As I mentioned, the cell culture system, we may also have some chance to develop neutralization assays once this is established. And of course, look at host responses that are induced upon infection. And with this, I want to stop here. I hope we can contribute in the future to this middle part of the disease, where we really need to learn a lot more. And I thank all the people and funding that were involved here. Thanks a lot.
Thank you, Volker, for this very interesting, very broad talk. Also bringing back some memories of when we were students in different labs across Europe, and who would be the first one to make a coronavirus clone. Some of these systems are still in use, because the MVA-based system is still in use. The back system, I think, is still in use, but it's nice to see some evolution, which takes now 2 weeks compared to 2 to 3 years. I mean, 20 plus years ago. Any questions from?
Hi, thank you for your presentation. It's mostly out of curiosity. You mentioned that there's going to be follow-up until the end of the study. How are you following up, and what are you trying to get out of the follow-up? Is it just to see if the cats are still healthy through a phone call, or are you looking for something else?
So of course, the follow-up, the ideal thing is that nothing happens. But we will particularly be interested in those cats that worsen again. And then we would particularly be interested in sequencing the virus. So is the virus still the same as it was in the beginning, or are there some changes? So these kind of questions. So to look a little bit more virus-centric, and to see what the virus is doing under these circumstances when it may come back.
So a question, we're still quite good with the time. Julia.
Sorry, Volker, it's a bit of a boring one, but what are your ethical restrictions in Switzerland for recombinant reverse genetics then, for gain of function?
I mean, there are no restrictions except that we need permissions. So in fact, you have to apply, of course, for those experiments. This will be reviewed. So in Switzerland, we have a national system. So you apply, and then there are certain federal offices that are consulted to give responses. There's also a expert committee for biosafety in Switzerland, a federal committee that is overseeing all BSL3 and 4 applications. And then at the end, you get the permission or not. Of course, the submission or the application contains, a risk assessment, all these points.
So I'm just thinking of provocation, right? Because part of the network, we have to advise potential policy areas and so on. And because of the situation in the States, people are now nervous in the UK because people are nervous in the States, blah, 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 blah. In your head, what do you think should be allowed in reverse genetics, and what are your red lines and grey lines or okay lines? Because you're the father of, and Zoltan too, kind of the fathers of reverse genetics for coronas. So
I think there are several layers. People are scared about biosafety. Let's say you create a virus, it escapes from the lab. Of course, the first thing you need to make sure is that the lab is safe. So that is not only a task of the institution, but also the regulatory authorities have to monitor, inspect, and watch that this is at the best level as it can be. And then, of course, the question whether you generate a virus with other properties, as you mentioned, gain of function. So here there's no proper line, and I also cannot tell where this should be. I think I think we should go into a discussion how we deal with that, because certain experiments really make sense, so they give us a lot of information. And the question is, how can we allow that those experiments can be performed in the best and safest way? Yeah.
It might be worth expanding on this later on in the discussion session that we have planned, if that's okay.
Yeah.
Because I think it's something that's...
I mean, you're right. I mean, the discussion is driven. I mean, I think it came up very strongly on Twitter or X.
Yeah, I don't have social media, so...
And there's a very loud group of people that is aiming for restricting everything.
Right.
Shutting down everything, essentially. But there is also, I think, mainly also in the US, on the political level, there are forces that support these ideas or are in favor of those ideas. And the question is how we as scientists think about that and how we exchange with policy how to move forward from here.
Yeah, thanks, Volker. And lovely work, by the way.
Yeah.
So there's been lots of research on coronaviruses that despite all of these decades of creating different recombinants, all the tools that we can use today for, as far as vaccines, I mean, still the conventional non-GMO modified live vaccines. So there's still room to develop. Lucy?
Yeah.
Thank you. That was really interesting. I've got a slightly more clinical question. It's really great to have the opportunity to talk to somebody who's really thinking about these things at a molecular level. When we treat cats with antivirals and then they come back to the clinic with FIP recurrence, I guess, do you have a sense of whether you think that is a reinfection, or do you think the virus is somewhere in the cat still and is coming back from within the cat? My instinct tells me that maybe the cat is getting reinfected and has the same immune defect that is allowing whatever it is about macrophages to go awry, and you get another round of disease, particularly knowing that people get coronavirus multiple times. But do you think it's possible that it's going somewhere in the cat, we can't detect it, but it's dormant? I'd be interested in your thoughts on that.
Yeah, it's a very good question. I mean, if we sequence later on the virus, we will at least say, or can say, it's the same virus or not, apart maybe from a few mutations. So if there's a reinfection with a different virus, we would see that. So we could then exclude that. But the other two things, is it then the virus that is coming back again, and why? Was it hidden somewhere?
I mean, I'm kind of wondering, we haven't really looked in the bone marrow of these cats, and I sort of worry about that, you know, what we see with some of the latent
I mean, there's a
viruses.
classical experiment in coronavirology with mouse hepatitis virus. So you can infect a mouse with mouse hepatitis virus, and the mouse will clear the infection. And what people did then is, after several weeks, they deleted or took out the B cells. And then the virus came back.
Yeah. So there is an active suppression.
There is
of
a possibility
yeah.
that a virus can stay somewhere unchecked but silent.
Because that's my big worry from the kind of, I suppose, public perception of all these antivirals being used in veterinary medicine, that somehow we are creating a monster for later on, because actually we're allowing many more cats with a persistent focus of a potentially horizontally transmissible virus to survive where previously they wouldn't have done. So yeah, I think it's really exciting and important work. Thank you.
Yeah.
Thank you.
This is a very interesting question, and we did see in the FCOV23 in Cyprus, we did see a lot of reoccurrences after treatment, but that were because the proper dosage wasn't available at that point. People were giving medication, the same medication from the beginning, but the animal gained weight, so it was subdosing actually. So that's one reason. But the thing is, I saw in your study that in one of your trial with GS and remdesivir, you were saying that for 6 weeks to 12 weeks. Were you trying to see if the treatment duration would be less than 12 weeks? Because that's a major thing for clinicians. Due to the cost. And this, as you can understand, this is a very important question. And the other part of my question is, if you combine two antivirals, which is potent for mutation also, do you, the purpose is to lower the duration again of the treatment?
Yeah. You're asking the right questions, and of course with our few cats we have treated so far, we cannot really tackle that, but the intention or our aim is really to contribute to that. So, because as you mentioned, there is this black market, and we have the same issue in Europe, and cats are treated uncontrolled by something, and what we really want to contribute to find the best, time period where treatment should be given, and also maybe later on assess if a combination may be useful in also lowering the dose of the individual drugs. But we are not yet there. So for us, it's very simple. We want to get experience how to do those trials. But I think together, it might be good to align to get and exchange experience to eventually get exactly answers to your questions. How long do we have to treat, and what is the best type of treatment?
Thank you, Volker. So we're moving to the last talk. Or more?
Can I?
Yeah.
Thank you. That was quite interesting as far as I'm concerned, because and the question asked before with the virus hiding somewhere, usually viruses hide in immune privileged sites. They don't express any proteins, so the immune system doesn't see them. The experiment that needs to be done there, and it's shouting for it, is one of these recovered cats to be immunosuppressed using cyclosporine and see what happens, if that is ethical, of course.
I mean, that touches one thing. So one basic problem we have also with our studies, experimental infection, essentially. Nobody wants to do it. But this is, of course, a big thing also if you, an important thing also if you want to assess later on vaccines. So I don't see anybody, or I don't know anybody who wants to do those studies, but...
we have needed to do that.
Yeah.
Because they're clinical cats, and some of them develop diseases after they recover from FIP and need to be immunosuppressed, and we haven't seen any recurrence of FIP in those cats.
Yeah.
Maybe last question, then we are moving because we're out of time.
I'll make it quick. So, as I understand, a lot of these drug trials are done on very well taken for clinical, well-managed cats. But is anybody looking at a shelter cat, which would be naturally immunosuppressed, and is that worth considering to do a shelter-based study to do this sort of naturally versus with cyclosporine?
So you mean?
Just a naturally shelter cats.
Yeah.
Better success than I do.
Okay, thank you very much, Volker, again. So.